Its arm
You
betrayed it, though.
I got it.
It's what?
Its arms betrayed it.
Oh, it's arms.
Okay. And
Those
I need to rotate
arms.
my butt.
Do you think he's reaching for something, or why do you think they're so long?
So in bucket 1, canister 1.
Yeah, they're definitely reaching for something. We often find them on corals, so they probably try to maybe catch things in the currents as well, because you will see them on corals very often on the colonies. And they often do that when you get close to them with an ROV, so I don't know if it's some kind of attempt at intimidation or something.
That would make sense, yeah.
You able to get it, Mike?
Yeah, I'm just trying to find the right angle.
Yeah, so the organism, or I should say the organisms that we're taking a look at right here, these are, there's a piece of coral here. I forget, is this considered a black coral?
No, that's probably a bamboo coral.
Bamboo coral. And on the bamboo coral, you can see we have a squat lobster. And so there is some interest in collecting this squat lobster, so that is what our team is currently gearing up to do.
Oh, why did that let go? Okay.
Do you know why they're called squat lobsters?
I mean, they are, in a way, squatters. I don't think that's the reason why they're called that way. You should ask an English speaker person here.
Why is that not locking?
Yeah, that's a great question. Yeah, on NA153, we called ourselves the squat lobster crew. And at the end of our expedition, we all took a picture like a squat lobster with our arms up in the air. It's really fun.
Okay, got the grip lock on.
Okay. Come on up to it.
Here comes the slurp.
Okay. Ready?
Yes. Okay, go ahead and come off for a second. Okay, off of it. Okay. And go ahead and come on.
Come on, squat lobster.
Okay. Okay.
There.
Oh, there he goes.
Okay.
Great job.
See in the bucket?
Almost.
There
it
it
is.
Okay.
Nice,
Slowly
Mike.
zooming out.
Very nicely done.
Thank you.
Now, should I change buckets so it doesn't jump out of there?
Yep.
Okay, go ahead and go.
Okay.
Okay, I'll go ahead.
Go ahead and stow the arm.
Yep.
That was bucket number 1.
Is that a sea spider on the rock just above it?
Oh yeah, whoa. Or could it be like a really
Thin
like
sea star?
brittle star?
Yeah.
So there is a brittle star here on the right on that rock. The white star is a brittle star.
Go ahead and zoom video.
All right, zooming.
So delicate.
All right, arms stowed.
Okay.
Okay, that's good, I think. Thank you.
Okay, go wide.
Going wide.
So let's just have a quick look at that sponge as well.
On the, on the
Behind the
left-hand side.
coral, yeah, behind the coral there's this stalked sponge.
Yep, no problem.
Let's just have a look at it.
I can get closer. Just give me a second.
That looks kind of one of these stalk sponges from the genus Hieronyma, and this is for a glass sponge, which are actually specific of the deep sea.
So can you tell us why they are called glass sponges? Are they made out of glass or something similar to glass?
No, sponge actually are made of like silicate, silicium. They have this sort of like, not really a skeleton, but it's called spicules. These are little like sort of needles made of silicate. And that's not just characteristic of glass sponges, all sponges have that as well.
Okay, go ahead, zoom video.
Zooming.
Do I have all my lights on?
You don't have mid on. You can get that on too. Mid's coming on.
Mid's coming on?
Yep.
Helps a little bit.
That looks good, thank you.
Okay, zoom out.
Zooming out.
All right, let's
Okay,
continue.
can I get you to porch in?
Roger.
Porch in. Porch coming in.
So then if sponges have like silicate needles, how come there are some sponges that people like actually bathe with and like they're actually like spongy and squishy?
Yep, porch is in.
Okay, thank you.
Well, those are not necessarily like super, I would say, like they can break easily, and so some of these, they don't necessarily hurt you, that's what I mean. Like some of the sponges, if you take them, they will actually hurt you like needles, but others, it doesn't do that. So these are the sponges that, for instance, you use to, I know we use them to paint the walls sometimes, or like other reason, like natural sponges, and those like break off and don't like hurt you.
Okay, so they're just more flexible or
Yes.
more brittle, I guess. Okay.
So that looks like maybe a mushroom coral that we see. If we zoom in, I'll be able to be
The
sure.
red one?
Yeah, the little red.
Yep, don't worry, let me get closer.
Wow.
Okay, go ahead and zoom video.
Zooming.
Looks like there is actually maybe 2 of them. And this is just another type of soft coral. We also call them mushroom coral sometimes. Maybe Anthomastus. I don't know if that name changed again, but there's 2 of them.
Can we get a zoom in with the cinema camera?
You don't have control of that back there?
No, not back here.
We don't believe have it. Up
I have it right here.
Do you? Okay.
Thank you. Perfect, all set.
Okay, Going wide.
Going wide.
Thank you for that.
On the her
Can
controls.
continue.
Beautiful.
Okay, we good here?
Yes,
Continuing on.
We're moving bearing 270 pilots.
Okay. Looks like there's a little something right here.
Oh, yeah.
yeah, there's something coming up ahead of us. There's another sea cucumber.
Oh my God.
Actually,
Honestly, I've lost
no,
count.
that's
I'm having a great day.
I think we've seen over 10 at this
yeah.
point.
I'm not sure what this is here from here. Let's see.
Watch out, please.
Watch out.
It's good.
I'll stop.
There's also this.
Okay, go ahead and zoom video.
Zooming.
No. Okay, we can zoom out. And there should be on your left another unbranched coral that looked a little bit different than the one we've seen earlier.
Okay, no problem.
Was that another sponge that we just looked at?
Not sure, a piece of something.
Watching.
It was not a rock.
Watch coming in.
No, actually that is not a coral.
That's good.
Okay, I'll stop.
Ooh, oops.
Boop.
Just gave it a little bit of a nudge.
Another boop.
No, that. Oh, that's broken. I think that maybe a sponge.
It looks almost flat, yeah?
Yeah, I think we bumped into it. Maybe it's like, yeah. All right, moving on.
Okay.
Yeah.
Watching.
Watch out. All in. And I'm getting pulled off just a bit.
Yeah, I know. I'm trying to get back over.
You have train peeking in too.
Oh, it's not.
Some more coral. Here.
Sorry, can we have a look? I guess we can have a look at both. They might be the same.
Okay.
This one might be a primnoid.
The one on the right?
Both.
Oh.
Okay, go ahead and zoom video.
Zooming.
Getting pulled around.
Yeah. Trying to get closer towards you.
Yeah, I think this time this is a primnoid, maybe some kind of Narella. You can see the polyps pointing down. And I think the one that we see in the back is the same.
Just take them
Yeah.
out of heading off.
We can zoom out, and there's another one that we can check in the background.
Okay, go wide.
Zooming
And
out.
I think it might be also a Narella, it might be the same.
You talking about the big one on the left or the one on the right?
The one on the left.
Okay.
Yeah, behind that rock.
Yep, going.
Yeah, I'm waiting until we get a bit more scope on the tether.
Is the boat all stopped?
Might just pulling you off.
Okay, go to Zoom video.
Zooming.
We've only got about a minute here. We gotta get going.
That one also has a squat lobster.
Oh, yeah.
Yeah.
Now that you pointed it out.
Can we also get a zoom on the cinema camera?
Cinema. Okay.
Perfect. Thank you.
Yep. Is that?
That's perfect.
Okay.
Is there any chance we can sample that, or you say you need to?
All set with cinema camera. Thank
Push
you.
it back if we
Yep.
need.
Coming back.
Oh, okay, zoom wide.
Going wide.
Okay.
Is
Do we want me to
also-
step back a little bit?
Uh,
Are we okay with this ship position? There might still be some pendulum swing.
yeah, there's still some pendulum swing. I think we can make it.
Okay.
Let me just get a position that we can.
And we're also interested
Watch out.
in the crab
Okay,
to
port's
have like
coming out.
host plus associate together would be
What?
great.
Yeah, you gotta grab the suction sampler.
Okay.
Good.
All out. Okay.
I'm guessing to more understand those relationships, correct?
Yes, correct. There's a lot of animals living on these corals, but we often don't really know much about
They want the
what are the
suction?
species associated with them. Is that species-specific association, and what is also the nature of this relationship? So when we can, it's great to have both.
Do you typically get those kind of samples, or are those rarer?
It depends. It can be difficult because let's say we try to sample the coral and then the crab might go away.
Atlanta's pulling me around.
Yeah, I don't have.
So it's
auto
often
heading on, so I'm
challenging, but
yeah.
we try when we can do it.
trying to give you everything.
Of course, of course. That's wonderful.
I can come down a little bit more if you
Yeah,
want.
come down a little bit if
Yeah.
you can.
Coming down.
Terrain is 5-10 meters off.
I'm 5, yeah. 5 meters, 4
Oof.
meters.
Okay. See that.
I'm gonna come up just a hair. 6 meters. Okay.
I don't know if I can get, if you can grab the suction sampler in that position. I don't know if we're going to be able to sample this. It's just not a very good position.
Okay.
Do you think we could do an eDNA sample here with the Niskin bottles? This might be a good spot to
Yeah.
do that.
Yeah, we could do in this
Yeah,
bottle.
we haven't done any of them yet, so let's just do one bottle.
Okay.
At 5 meters off the deck.
Yep. I'm just going to sit down so you can.
Okay.
What bottle number would this be?
Should we do bottle 6?
Okay, Niskins.
Terrain is 5 meters off.
Yep.
Can you give me Niskin in the camera?
Yeah. Why is that not? Well, that's not it. Yeah, 1 second.
Wheel.
All right.
So for our viewers at home, I believe if you look on your feed at number 3, you can see those Niskin bottles we're talking about on the side of Hercules.
So we're going to do 6?
You got the small cameras?
Yeah, I see it.
Okay. And I can zoom in if you want.
Back the camera back.
Camera is retracted all the way. Yep.
Can I get you to turn the camera?
Yeah, main camera?
Yep.
Yep. Okay.
Perfect.
Okay, I'll stop.
Okay.
Hayden, is that sample 10?
Okay, that sample went. Okay, turn the HD.
Thank you.
Okay, go ahead and coming back on the HD. Is the knife tapped out? I might have accidentally touched that when I was grabbing.
What happened?
The knife, I think it might have slipped out just a bit.
Okay, I'll see if I can.
Sorry.
Not the knife.
Train's a couple meters out.
Yeah.
There you go.
4 meters off the bottom. Nice.
So what just happened there with those Niskin bottles? We just took a sample of eDNA.
Scoring you back up.
And eDNA stands for environmental DNA.
Maybe. There it is.
As you're watching our dives, you might notice there is all kinds of this white floaty stuff in the water, and that's known as marine snow.
Porch in.
Okay, porch coming in.
And I'm going to get closer to you.
Yeah, and let me come on up because I'm at 4 meters.
I can add a little bit to that. This eDNA is pretty useful
okay, porch
too.
is all the way in.
Because we can figure out
let me know when I can come up.
possibly from those marine snow particulates what organisms are living here that we don't see, and that's pretty massive.
Yeah, so it's a non-invasive way to get a sample of the different life forms that are around this area. So as we trigger that Niskin bottle to open, water comes rushing into that bottle, and we bring it on up to the ship, and it is then further processed so we can get a biological profile of what kinds of animals are living in this particular spot that we are observing currently.
I got to get we're tail to tail right now, so I got to get back where I need to be.
I'm going to do a slight push in just for the vignette. Thank you.
Am I good to come up some more?
I'm watching you. You look pretty good. Almost even.
Yeah, coming up just a bit. Try and get a little bit more space.
I'm just trying to get back to you.
Yep.
So we had a question come in a while ago asking about the main aim of this expedition.
Okay, I'm at 12-meter altitude.
So each expedition has different objectives. The expedition
So
prior to
auto heading back on?
Yep. Coming back towards you.
prior to NA174-173, that expedition definitely had more of a historical, archaeological focus. And then this one here, NA174, we are aiming to get pretty much baseline data about the biology as well as the geological makeup of this area around the Marshall Islands.
Okay, that's much better.
Yeah, a lot of this area that we're in is pretty much unexplored, and so it's around 300 seamounts, correct, that we're looking to explore, hoping to explore.
Okay.
Yeah.
Okay, you can get the ship moving.
Roger.
Yeah, so this area specifically is just very, very much largely unexplored, and that is why it is very important to be doing this type of work, especially because it's in such a pristine area, and around these waters there is a newly designated marine sanctuary. So to help to uphold that sanctuary status as well as to support further policies and protections, we definitely just want to get as much information about this area as we possibly can. So that's definitely the aim of these expeditions as far as NA174 goes. We're trying to see if we can just get a better idea about what things are living down here and also what does the geology of this area have to tell us.
Are you good for me to go ahead and turn off your port and starboard light, or do you want that on?
No, you can turn it off.
Turn it off?
Yeah.
And then Fanny, there was a question about sampling. How do we decide which samples to take versus what not to take? How do we make those decisions?
That's a very good question. I mean, there's a variety of factors, so it depends also on our respective research project, things that we may be interested in. So for instance, for what I'm doing, there's some families of corals that I'm particularly interested in looking at. And then anything that looks unusual or new compared to what we've seen in other parts of the Pacific is also very interesting to sample, especially if we suspect that this may be a new species.
And then as we just saw a few minutes ago, we did, or we were attempting to make a collection with a squat lobster and a piece of the coral that that squat lobster was hanging out on. And so that's what we call an associate.
Close to, looks like in the sonar. I'm fairly close to something.
So I think we're going to turn
right in front
off
of me.
4 and 5 and start 6 and 7. Can you get that? 4 and 5 off, 6 and 7 on.
Just take a look around real quick.
4 and 5 off, 6 and 7 on.
I do have a ridge off
Yeah.
dead ahead of me, so I'm gonna come up.
Somebody was asking, does anyone on the team think Megalodon is still alive? I love it. I don't know. I haven't asked everybody on the team that question, but
Can we have a quick look at this here?
Yeah, can I get a porch out?
Porch coming out.
maybe that's something I could do tomorrow during breakfast.
Okay, video.
Okay, zoom in.
Yeah, that looks like another black coral, similar maybe to the one that we saw, but different color.
Pump 4, 137407. Pump 5 is
All right, we can zoom out.
141447.
All right.
Both are milliliters. Okay, zooming out.
Are you happy with that?
Yep.
Okay, zoom out.
Ready to keep traversing?
Yep.
Tether's about to come tight.
Okay, pull it in.
Porch coming in. All the way.
Yeah, so there are several different techniques that are available to capture specimens or to take samples. Hercules, the main ROV that we are using on these dives, does have 2 robotic arms. So it is able to use those robotic arms to pick up certain items as well as take small samples of maybe a small fraction of a coral that we saw just a while ago, about an hour ago. And it does have some bio boxes on the ROV too. So we are able to put whatever samples we do collect within those boxes. I like to kind of say it's almost like Hercules' pockets.
Good, we're on a summit.
Yep.
And then in addition to that, we did just see some eDNA action happen a few minutes ago with those Niskin bottles. So there are many different ways to do some sampling while we are at these depths. And then when Hercules comes back up onto the ship, our science team then takes those samples and they get processed within our data lab, or excuse me, our wet lab. We do have a wet lab on the ship, and that's where those samples are processed and stored properly until we get onto shore, and then they can be analyzed further.
Is that still an issue if we're not in any channel talking?
Can we have a look at this coral coming up ahead of us, please?
Yeah, can you all stop the ship?
Looks like there might be a crinoid on it as well, maybe. So that's a bamboo coral. That's a bamboo coral with a
Kaden, are you talking to me?
crinoid attached to it.
Okay, go ahead, video.
All right, zooming.
You can really see the skeleton with those like black knots. That's why it's called bamboo coral. And this skeleton looks exposed and overgrown. Yeah.
Can we also zoom in with the cinema camera as well?
Cinema cam coming in.
Thank you.
Yeah, you can see
Let me
that
know
coral has exposed skeleton, and you have like other organisms overgrowing it. Looks like some kind of maybe hydroid or something on it.
So that's max on the cinema cam.
Perfect. That's good. Thank you.
What do you think happened to the coral
set.
so that it has those spaces that are bare?
All set on the cinema cam. Thank you.
Well, that coral is exposed skeleton means no more live tissue, so live tissue, so that means the coral is dead. I don't know what happened to it, but then the skeleton remain in place even after the coral is dead, and so that's why you end up having other organisms that use it as a substrate. All right, we can zoom out.
The wide video.
Going wide.
I don't know if I've ever seen a crinoid that really deep, dark purple. That was beautiful.
Yes, it was. Can we continue on?
Yes, we can.
Fanny, you might have a better way to answer this question, or a better idea. Somebody's curious to know what kinds of larger sea creatures might we discover or might we find on a dive like this?
It's hard to tell. I mean, the largest animals in a way that we might see probably are some of the corals, or some of the colonies can get pretty large. In the deep sea, most animals actually tend to be smaller at deeper depth, mostly because there's just not that much food available. Although we do observe what we call gigantism for some animals. Sometimes sea cucumbers can get pretty large, for instance. But there's not going to be extremely large animals at this depth. We might see some fishes, but not giant, especially around this 2,000-meter depth. So I would say probably a coral are some of the largest that we may be able to see.
Yeah, so following up on what Fanny was saying, these animals that do live in the deep sea are living in a very, very extreme zone. So there's extreme pressure, there's not much food around, so it's not very conducive to being able to grow to be very, very large. So a lot of the animals that we will be seeing will maybe be on the smaller side, but we never know. But there was a sponge that we did see a couple of years ago in 2023 that was easily twice the size of our ROV Hercules. And if you're wondering how big Hercules is, it is about the size of a USPS mail truck. So it was a very, very enormous sponge that we did see in the depths. So yeah, definitely some sponges can get to be quite large, and as Fanny was saying, probably some coral species too. And yeah, there's a very interesting question that came in about light in the deep sea. So yes, indeed, the deep sea does not get any light whatsoever. So our presence in the deep sea. Hercules, you can see, has lots of really amazing flashlights. And if there is an animal that is around that does have eyes and can perceive light somehow, it will absolutely be the first time this animal would be seeing some light. So it's really, really interesting to think about.
Yeah, it looks like some sea urchins here. There's another one at the top as well.
You guys interested in looking at it, or just keep going?
Let's zoom in on it just to have a good image of it.
Go ahead, video.
All right, zooming in. I'll zoom a little more.
There's a little brittle star too.
Look at the spines. They're almost see-through. It's crazy.
Yeah, I think that's good. We can zoom out and continue. Thank
Okay,
you.
zoom on.
All right, zooming out.
So we want to try to reach the top of that ridge, and hopefully we can find some flatter areas where rock may accumulate.
Okay. We can keep moving.
It looks like a palm tree.
Yeah, it kind of does.
It's close enough.
Just about. Fanny, that sea urchin got me thinking. I'm just totally curious. Are there any, or is there any evidence of there being any like poisonous or venomous deep sea creatures that we know about yet?
Yeah, good question. I'm thinking, I am not actually aware specifically of like poisonous species, but I wouldn't be surprised if there is.
Yeah. Well, I mean, okay, so technically coral are cnidarians, right? So do the deep sea coral species, do they technically have stinging cells, or are they stingerless?
They do. I mean, not as much as a jellyfish, for instance, but some of them do. These like gorgonian corals, octocorals that we saw, not so much. So if you touch them, you probably not feel hurt.
Yeah.
But some corals can have like a lot of stinging cells, but not these ones specifically.
Okay. So right now we are using visible light on ROV Hercules so that we can be able to see what is down there in the depths. But we do have somebody who's curious to know if we
can have a look on the right side maybe. I don't know if there are any rocks over there.
Yeah.
There's another dead bamboo coral.
It almost looks like there's something on it.
Yeah, it's another crinoid, yellow one this time.
It's crinoids really like those dead coral or dead sponges. Do you think it's because they have like a higher vantage point if they're,
Yes,
yeah, that's
because
why.
they're suspension feeders as well. So being on a coral gets them better access to any kind of food that would be in the water column. So that's a good spot for them to be on. Yeah, we can keep going.
So yes, we had a viewer who was wondering if we ever use like infrared light on our cameras with Hercules or Atalanta or anything like that for exploration. Do our ROV pilots know anything about that?
Infrared doesn't work very well underwater.
Say more.
The refraction of the spectrum does not protrude very far from the vehicle.
Got it. And especially because we're in the deep sea, right? Like, aren't most of the animals like red in color for that reason or something like that?
Benny, do you want to zoom in on this?
No, we're fine. We can
Okay.
continue.
I'm getting a weird reading in the Atalanta sonar, but I got like nothing up ahead. So I don't know if it's just reading the edge there.
Hopefully it's not an overhang.
Yeah, I know, that's what I'm like slowly starting to look up. I don't see anything that close though. We do have a pinnacle off to my port side. Yeah, about 9 meters. Yeah, it's coming up on my port side.
Yeah.
Okay.
Just got really steep.
Yep. Coming up. It's starting to beat you a bit. Okay.
Could we have a quick look at this ahead of us?
Can you porch out?
Porch out. That's all the way.
That one is probably going to have also his own little squat lobster. Looks like some kind of Keratoisididae. With this angle, it's hard to tell
Okay,
which
zoom
one
in video.
is.
Zooming.
Some kind of Keratoisididae. Look at that.
Got danger close.
Yeah.
Really steep.
Yeah, looks like a Keratoisididae.
So fluffy.
And you can see it has his own little squat lobster. There's two of them.
Yeah. Aw.
They're almost see-through.
Look at that.
Danny, I got structure fairly close on my port side.
Okay.
Yeah.
Coming up.
So that's also called a gold coral, and you can see
Sorry.
why on this image.
Do you want me to get the ship to move
It's
away from there?
golden.
Yeah, if you can. If
Okay,
we can get
let's
it to
step
move
back.
back towards you.
Yeah, bring the ship back.
So, Danny, do you think it would be possible to cut like a piece of that coral to also get that lobster like somewhere here? To get one of these lobsters as well, or is it tricky from here?
Yeah, we can, but let's wait a few minutes. We've
Okay.
got a mountain on the side of Atalanta.
I'm coming up.
Look at like all the little organisms around it. Like they're like flies. That's really neat.
Oh yeah, it looked like little sea gnats or something like that.
Yeah.
I'm clear-ish, but not
The
by
ship's,
much.
well, the ship's coming back towards
Yeah,
us.
yeah,
Good catch, Mike.
yeah. Okay. Just keeping an eye on it.
Yeah.
That came up quick.
That's what always happens. So you want a piece of this coral with the squat lobster on it? Is that what you were saying?
Yes, that would be great.
Okay. Let me make sure I holding in position here.
Do you want me to hold ship position or let it keep moving back towards Herc?
Moving back towards Herc is fine.
Okay, roger.
Danny, do you want to zoom out?
Yeah, zoom out a little bit.
Yeah.
That's good. Am I getting drug around by Atlanta? Is that what's going on?
Probably. I'm at 22 meter delta from you,
Yeah.
but I'm...
Still over top of that mountain.
Yeah. It's...
Okay, let's just wait a minute.
Yeah.
Let Atlanta swing back over top of us.
Yeah, we're just starting to get towards our goal. Is it okay if I do a DVL reset?
Yeah, go ahead. Video, can you go ahead and go wide?
Absolutely.
Yeah, you're pulling me.
Sorry. Yep. Thank you. Sorry.
We're closing in on our goal.
Yeah, Atlanta just pulled me off the rock.
Yeah, sorry. I can't go down lower.
I know. Fanny? Fanny? We're not going to be able to just collect that sample.
You can't?
No,
Okay.
I'm sorry.
That's fine.
All right, we'll continue towards waypoint 7.
You want porch in?
Yeah, go ahead, porch in.
Okay, porch in.
Hey, Isabella, you just did something called a DVL a few moments ago. Somebody is curious to know what that might be.
Yeah.
So hold on 1 second. Before you take that step, I'm still pretty close to.
Yeah, don't
Yeah,
move.
let's not do that.
Yeah, Atlanta just ripped me off the mountain.
Let me know when I can start coming back up, Danny.
Go ahead and go up.
Okay, coming up.
Are there any other cool questions coming in?
Yes, I want to let the ROVs get situated first, and then we'll go ahead and dive on into those questions.
Yep, good idea, good idea.
All right, I'm feeling a lot happier.
So we have another bamboo coral, black coral, sorry. And then, yeah, there seems like to be a few things right here, if it's possible to have a look at them.
Yeah, we can. We're getting out of the danger zone, so I think we'll be fine.
Yep. And some other sponges here.
Still something close by.
So you see the difference in color for that one?
Yeah, absolutely. The other one was looking kind of rough.
So the black coral is the smaller one here in the corner. Maybe some kind of patties or something similar.
Okay, video. Go ahead and zoom.
All right, zooming in.
So we have, seems some coral that is being overgrown by what we call, so that is some, yeah, zoanthids maybe overgrowing that coral. So the coral is the pink one.
Wow.
So it's probably some kind of Tharagorgia maybe.
Why don't we do
And it's completely overgrown, and there is a brittle star here, and Niskin.
Yes, I think it's a good spot for Niskin.
Okay, we could do a
Variety of
Niskin.
things here, yeah. Let me know what bottle number you do.
We did 6 before, we'll do 5.
Amber, how do I get to the bio? Niskin 5? Sounds good.
Okay, can you go ahead and zoom wide? And can I get you to back in? Video, zoom wide.
Zooming wide.
So Fanny, are these like parasites on the... All right, so definitely not associates. Those are parasites. Whereas it seems like when we find associates, those are more...
Okay, can you turn the camera to the Niskins?
Yeah, panning left.
Associates are just kind of hanging out, whereas parasitic relationships, that is a negative impact on the host.
Okay. Yeah, 1 second. Let me check with Danny. Danny, are you good for me to change dive salvo to sample?
I don't need sample.
Okay.
I got it.
What were those parasites called again?
These are zoanthids. They are kind of related to anemones, and we find them often overgrowing other corals. That coral colony is almost entirely overgrown. But it still has its brittle stars, which often these brittle stars are found on corals as well. It's very common.
3, 2, 1, Niskin.
Closed.
Okay, so the zoanthids were the smaller, tiny,
pan right.
tiny like anemone-like animals, and then the brittle stars are the larger red creatures that we can see kind of hugging the coral.
We're going to do a slight zoom in.
Maybe before we go, if we can zoom in just on that black coral over here, just to have a good image of it, and then we can move on.
Sounds good. Go ahead and zoom.
All right, zooming.
Somebody earlier was asking why are they called black coral if they don't look black. I believe it's because their skeleton is black. Is that right, Fanny?
That is correct. And if you really look sometime in transparency, you can kind of see the black skeleton. And actually some black corals are being collected to make jewelry. So the skeleton can be used for that. And there's still a commercial harvesting occurring in certain states and certain part of the world for making jewelry.
Really?
All right, I think we're good with that coral. We can zoom out.
All right, zooming out.
Go ahead, zoom wide.
So then are there shallow species of black coral?
Okay.
Okay, you can extend
Sorry?
the camera.
Are there shallow versions of
Yes.
black coral?
Yeah,
Okay.
you will find black corals in shallow waters as
I
well.
see. I was like, that's a lot of effort to go to the deep sea to get black coral. Okay. Very interesting.
Okay, taking off.
Still have some pretty close structure just in front of me.
Yeah, we're gonna get up over it. Yeah.
Beautiful sponge there.
Haven't done a ship movement, so you guys just let me know when you're ready.
No problem. Are you above the structure?
Looks like I am above the structure now. Yeah. Yeah, it's off my radar. I'm starting to see you come into view.
Okay. Okay, we can go ahead and start the ship move.
Roger.
Wow. Are all of these crinoids?
Do you want to look at anything here?
No, I was just looking for rocks mostly, see if there's anything that looks like is movable, but I'm not seeing anything right now.
Okay.
Brittany, I'm kind of able to answer a little bit more about the DVL now. So
Please do, that'd be
minutes
great.
later. Okay.
Thank you.
Yeah. So DVL stands for Doppler Velocity Logger, and it is an ADCP. So ADCP stands for Acoustic Doppler Current Profiler. So that is used to kind of understand how fast and the direction and particles in the water are going on. That's an ADCP. So the DVL kind of faces downward on the ROV, and it's kind of on like the aft bumper of Herc. And so the DVL doesn't know exactly where it is, but it's measuring the Doppler shift of the seafloor below it. So sometimes you'll kind of hear us talk about like Doppler, we have 4 beams, and that's kind of the 4 beams are how we know kind of where the seafloor is, that we're getting that return. So the DVL is communicating with NavG, NavEst, which is one of our kind of navigation programs over here. And so over time, as we keep taking these different steps with the ship, we're having like, we're kind of understanding origin and we're trying to create a path from that origin, but it can get kind of lost over time. And so it's precise, but it's not accurate, is how it's usually described.
So if you see, sorry, that coral here, and there's a few rocks around, if we can just poke around and also have a look at that coral. Sorry for cutting.
Sounds good.
Isabella, that was really interesting. Thank you.
Yeah, thank you.
Are we moving the ship or? Okay. You're starting to get tugged around. Okay. Come down.
Yeah, I don't know if that's an acceptable rock or shape, but maybe poke that one here. That coral is definitely alive. Looks
The
like
more
a-
angular the rocks, the better. The really rounded ones here might be manganese nodules.
Okay. So that's another bamboo coral. Live one, this one. So maybe the skeletons we saw were from that same species.
So you want to try to get that rock?
I don't know if it's the right shape for that rock.
Okay.
Apparently,
I'm not sold on it. Something a little more triangular with really
Okay.
sharp corners. There are a couple of those, but they're behind us now.
okay, so yeah, something more like triangular shaped. That one is a bit too round. So if we see anything that looks like will be movable,
Okay. Do you
yeah,
wanna
it's
zoom
like
on
something
this?
with an angular shape, not round, basically.
Do you want to zoom in on this coral, or are we good?
Yeah, since we're here, let's zoom in on it.
Go ahead, video.
All right, zooming.
Look at that.
Beautiful.
Look at that.
These corals always remind me of candelabras, the way that they're shaped.
Well, that one apparently is called Tridentices candelabrum.
Oh, well, look
So
at
there
that.
you go.
Would you look at that?
Good description.
I guess so.
Yeah, exactly. All right, we
Okay.
can zoom out.
Zoom wide.
All right, going wide.
And shrimp.
Shrimp.
So yeah, maybe let's just look around in that general areas to see if we can find any angular shaped rock that seems like could be movable.
Oh, the shrimp make me laugh. I'm sorry. They're funny.
Their movements are crazy.
I know, they're pretty erratic. I mean, they're seeing this giant thing approach them from the abyss. I'd be kind of erratic too, I think.
Yeah, I think that's pretty fair.
It's fair.
It's like aliens, right? You know, coming down and abducting things.
I think they're kind of cute.
Oh, absolutely. So there's another nav question that came in. This is kind of like what came first, chicken or the egg? But it's like, does the ship try to keep up with Herc, or does Herc try to keep up with the ship?
Sorry, I caught the end of my headset fell off.
Oh, wow.
Can you repeat the beginning of that again? My bad.
No, the question was, does the ship try to keep up with Hercules, or does Hercules try to keep up with the ship?
I guess they're all working in tandem. I don't know if it's like a chicken egg situation. I think it's just like they're all three connected to each other. So we move the ship kind of in the direction that we want, and then Atalanta is above Hercules, so Ata swings towards that direction that we took the ship movement, and then Herc Herc has more control of where it gets to go, so then Herc kind of wanders with Ata, but Atalanta has less control of its movement, so it definitely gets dragged by the ship a bit more, and then gives like situational awareness for Herc, if that makes sense. So I think maybe in this case the ship is comes first, and then everybody else follows.
Okay.
Can you start a midship move?
Yes. Speaking of
Of ship.
Yeah, so if you're looking at home, I believe on the viewer feed number 2, you can see the view of Hercules from ROV Atalanta. So Atalanta is in the middle of the ship, and it's in the middle of, excuse me, it's in between Hercules and the ship. So it's kind of decoupling the movement of the ship so it does not disturb Hercules. And that's why the video feed from Hercules is so nice and smooth. And you can kind of see that the Atalanta camera is kind of moving up and down a little bit. And that is because of that movement from the ship.
Yeah, and Ada has like a more open frame to kind of allow that water movement to go through it, so it kind of does bob up and down with the ship movement. And it has, it's heavy, whereas Herc is buoyant.
There we go, yeah, thank you. So we have now reached the 2,000 meter mark. So when we first started this dive several hours ago, our depth was 2,200 meters deep. And we are making our way up this unnamed seamount, and our current depth is 1,988. So still have a ways to go until we reach the top of the seamount. But there's lots of exploring to do, which is always tons of fun. And again, if you're just joining us, you are certainly welcome to write questions in the chat, write your comments. We do see them as they come in. And this is our very first dive of this exploration, or excuse me, this expedition. So this is NA174, and we are exploring the waters around the Marshall Islands. And our main objectives are to get a better idea of the biology of these waters, of the deep sea around this area. So what kinds of living things can we find in the depths? And then also we're interested in getting a better idea about the geology. So that's why we're trying to look for some rock samples that are nice and angular. We do have a amazing geologist on board who's looking to get those rocks so that we can get a better idea, a better story about what the seafloor looked like long ago and how it came to be. And ultimately, we're just trying to get all of this baseline data so that we can have a better idea of how to further protect this area around the Marshall Islands. Someone is asking if Herc drills or takes core samples. From what I understand, it does not drill into any rocks, but it is able to take push core samples in the sand or sediment. Is that correct?
Yes.
Cool.
Yeah, we have a quiver on our starboard bio box that has 5 push core push cores on it. So if we run into something that we can go ahead and push core into, we can take up to 5 samples.
Thank you, Mike.
What's that thing down there?
Yeah, I was wondering that too.
What are you looking at?
Center southern.
It's down here. It looks like a sponge.
Well, we've got a few minutes.
See right there at the bottom.
Maybe another sea cucumber, perhaps.
Oh, I see. And we can zoom in on it. Yeah, it looks like it, and with a crinoid on it. Yeah. There's 2 actually. There's another one here. So that's a crinoid, probably on a sponge. Yeah, and this is also. Yeah, definitely sponge.
Kind of looks like quartz. It's cool.
That looks crazy.
Yeah, it does look like quartz.
There's a smaller one right behind it. Looks like a lollipop.
Or like a dandelion.
Can you zoom out?
Yes,
zooming out.
Zooming out.
So cute.
All right.
All right, we can
Sorry about that.
continue.
Just thought it looked interesting.
It's fine. We're waiting for the ship to catch up.
All right, so again, our current depth is about 1,987 meters, and you are able to access that information if you go to nautiluslive.org, which some of you are viewing the live stream on that website. So if you are on our main screen on the right-hand side, you'll see where we have some of the data, and then at the bottom of the bar on the right-hand side, it says click here for more data. If you click that button, it'll take you to where you can see Hercules' depth, you can see the water temperature, oxygen. So if you are curious right now, the water temperature is a nice and toasty 2.27 degrees Celsius.
These are starting to look a little bit loose here, especially over on the left.
On the
just
left.
coming into the center.
So you see one in particular?
Not yet. Could we maybe poke a little bit?
So these rocks look a little bit looser, so we're looking for something that could be, that we could move.
Maybe we just went over one. It looked a little bit loose.
More in like the sediment area, maybe. It might be a little looser. We can maybe poke 1 or 2 just to check whether they move or not.
I'm coming up.
Bottom of the screen.
The potato?
Yeah, a little
Yes,
potato there.
but it's very
She doesn't want potatoes.
Yeah, we want an angular.
No potatoes. Oh. Damn it.
Let's stop pumps 6 and 7, and ending samples 6 and 7.
My apologies.
Thank you.
So we're pausing here to see if we can do a collection of a rock. We have our geologist in the control room with us who is making a selection of
Circling to the right upper.
which one she would like to collect.
Yeah, so maybe try to poke down one here, maybe a bit big, and then check also on the side.
Okay.
Data, did you need me to change the location of any of the samples?
So that one directly dead center?
Yes, that one here. That?
We can poke at too, but I like that one more.
Okay.
That one. Okay. Yep, thank you.
So all of these rocks that we're seeing on the seamount, they are covered in what's known as a ferromanganese crust.
Oh, it's moving.
Our geologist is very happy that rock is coming on up. No questions
Rock
asked,
time.
it's cooperating.
Oh, it's got something on it.
Oh, and it's got an associate.
It's like a brittle star on it. All right, side box.
Okay, you want to pull the bio box out for me?
You want the
You want the
or
toolbox
starboard?
or the bio box?
It's bigger.
Give me the starboard box.
In the side box?
Yeah.
Okay.
You want your sample salvo, or are you good with that?
I think I got it from here. I'll have you swap out my... Yeah, I'll do it. Go ahead and pull the box out.
Yep, coming out.
There we go.
Val is fighting the urge to do cartwheels in the control room.
Question is, will it fit?
We'll make it fit.
Oh
Bang,
boy.
bang, bang.
Ooh.
Ooh.
Yeah.
Perfect, great job.
Associate.
There
lucky
goes
job.
our pedal star, yeah.
Close.
Yep, go ahead. E.
E.
All right, so can
Alright,
anybody
box is closed.
explain why it was that specific rock as opposed to another bigger one or smaller one or more circular? Does anybody have an idea?
Yes, they don't.
All right, really nice. Thank you.
I think that the more angular the rock is, the more it was likely broken off from something larger. Whereas like the more rounded, potato-like shaped rocks are just not suitable for sampling. That's all I know.
Okay, I'll take it.
Okay, we
All
can get
right,
the ship
so
moved.
let's keep going.
Starting pumps 8 and 9. Did you get that?
Oh, it might. Why? Oh, quite a bit heavier.
Okay.
Did you get that we started 8 and 9?
Oh, the heavy rock.
Okay, thank you.
Taking your vitamins?
Thank you.
I'm gonna have to drop an Alvin right before we
Yeah, I was
ascend.
gonna say.
Like flying Lukai all over again.
Constantly negatively buoyant.
Yeah. Still dropping.
Yes. Pump 6 is
Okay, let's do this.
174424.
Let's biobox out.
Okay, biobox out. Starboard or?
Not biobox, sorry, porch.
Okay.
137851.
Yep, go ahead.
Porch is out.
Okay, you want to go ahead and throw one of those Alvin rates?
Yeah. Go ahead and pan right with the sky. And up. Okay.
Because that's 0 Z bias, and I'm just sitting on the bottom.
Jeez. Yeah. Okay, coming out.
It's still a little heavy, but it's manageable.
Is there anything you're looking at over here that's like?
Looking at how the vehicle is sitting on bottom with no thrust. So normally you don't have any thrust, the vehicle's going to want to float
Positively buoyant, yeah.
Okay, arm is stowed.
Okay, poaching in. I don't know who came up with the idea for these stream decks, but they are great.
Yeah.
Thanks, Dan.
You want your camera to go back to the port rail?
Oh, yeah, sure.
Let me know when you're ready to keep stepping.
Okay. I'm set on this side.
Okay. Feeling good, Danny?
Yep.
Well, looks like there's a really tall coral up ahead. Or maybe it's fine, I don't know. Something tall up ahead. Another urchin.
A little bit better.
Yeah.
So I think we're coming up another bamboo coral. Can we have a quick look at this?
Yep.
And that one is...
Oh, the sunset's pretty on the Atta.
I tried to put the sunset on the camera for the satellite 3 for the viewers at home. If they want to watch it.
Thank you, Kelsey.
Yeah. It's a nice one out there.
Yes, I don't know if you heard that. If anybody's watching at home, we do have the beautiful sunset happening right now on satellite feed 3 if you were interested in seeing it.
Do you want to zoom in on this coral, Fanny?
Huh?
All right.
Do you want to zoom, or are you happy?
Science. Fanny,
Yeah, that's good.
you good with this?
Yeah.
Okay.
Yeah, I don't think that's anything. Yeah, I was thinking about what is this? All right, let's continue.
I'll let her catch up.
Did you log that we dropped an Alvin rate?
I did.
Excellent,
Yep.
thank you. I'm like perfectly trimmed right now.
You know what?
I'm like perfectly trimmed.
What does that mean?
Pretty neutral.
Oh, okay. So dropping that weight really helped.
Oh yeah.
Just cruising.
Can we have a quick look at this right here? It looks like the same as earlier, actually, with the zoanthids, right?
Which one are you more interested in? Okay.
But I think it's the same Paragorgia colonized by zoanthids.
Go ahead, video.
All right, zoom in.
Let's see. No, these are different. Yeah. Yeah, I think that's good.
Okay, go wide.
Thanks.
Going wide.
That's the same thing. Another coral completely overgrown.
So the pink part that we were seeing, that was the coral, and then the yellow parts were the zoanthids. Is that correct?
So
I was asking if the pink portions that we were seeing, those are that was the coral, and then the yellow.
yeah, so the yellow is the zoanthids,
Yes.
and the coral is pink.
Right. You don't hear me? Look at that. So we have some people online who are curious about what it takes to become an ROV pilot. And Mike, the other day you and I had an interaction and you shared some very interesting information about video games. Can you say more about that?
I mean, yeah, definitely was asked the other day, you know, if video games help. I think those of us that do pilot and play video games, the hand-eye coordination is definitely benefited from it. So it's also situational awareness. So depending upon, I guess, the game that you watch or that you play. But yeah, no, a lot of hand-eye coordination, a lot of different screens that you're looking at and just constantly kind of glassing from one screen to the next. And as you can probably see on from our control van here, there's at least 20 different screens in front of both Danny and I at the moment. So
I just put it in the sunset cam in the corner so you can kind of see up on the, there's a lot of desktops along the front, and then the very back wall is what all the ROV pilots are looking at, so it's pretty full.
So one thing that helped me and would help most people in this situation of becoming an ROV pilot is you want to be fluent, you want to study some kind of engineering, whether it be mechanical or electrical.
When I'm speaking?
That's going to give you a leg up to becoming a pilot. But for
Danny,
me,
sorry, can we pan to the left, the camera? There's something on the left.
Yep, no problem.
Thank you. There it is.
Oh.
Sorry, I always feel bad cutting.
Wow.
So that's an Iridogorgia.
Oh my.
So beautiful.
What did you say that's
moly.
called? What did you say that's called?
Iridogorgia.
Iridogorgia?
Iridogorgia.
The same, yeah. It's
That's
super
beautiful.
beautiful coral, and these can get like really large sometimes.
Do you know why they have that crazy spiral shape?
Yeah, I am not sure why that shape. They're often very flexible. So if you have a lot of current, they actually bend a lot. So you usually don't find them in crazy current necessarily. But I'm sure that spirally shape probably has some kind of advantage, I would say related to hydrodynamics probably. Maybe that creates some kind of like microturbulence also, and that helps maybe gathering food near the polyps. So a lot of corals, their shape depends on the currents and are adapted so that they optimize currents and microhydrodynamic at the centimeter scale so they can optimize food capture.
That makes sense.
You can go ahead and zoom video.
All right, zooming.
And correct me if I'm wrong, these are also known as firework coral, is that correct?
On the ground.
I actually do not know the common name, but so it's possible.
Okay. I mean, it would make sense to me.
It's so pretty.
So pretty.
They're beautiful.
It's,
Wow.
yeah, that's amazing. That's not something you see every day.
We might see it every day here.
This would
If
make
that's
a
the
wonderful
case, I hope so.
This would make a wonderful screensaver for my computer.
Oh my gosh,
Yeah.
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 Easily.
Maybe that's worth one of our Niskins too.
You want a Niskin?
Yeah, let's do it.
Okay, that's
You'll get a
we can pick it up.
Niskin.
And actually, I would be interested in a piece of that coral as well.
okay.
Doing
Let's
it
do
all.
it.
Asking a lot.
Go wide video.
Going wide.
This is bottle 4. I
Give me just
assume.
go
I'll
ahead
do the
with everything,
Niskin
but
and
just
then
give
we
me
will
some time to
sample.
get this all down.
Sounds good.
Oh yeah.
Look at that.
I wonder how tall this coral is. Looks like it's like at least 6 feet, I'm imagining.
They can get really tall. I have a colleague who told me like once they ran into an Iridogorgia that was taller than the ROV. They were like
Yeah.
underneath the ROV.
Wow.
And then that makes me wonder how old it is. Because I know that things in the deep sea tend to grow slower, right? So,
Oh, we know like deep sea corals have pretty high longevities. From the few corals that we've aged, we can age at least several hundred of years. There are actually some black coral and gold
so
coral
this will be
around the
good
Hawaiian
Niskin?
Islands that have been
This will
estimated to
be,
be over
the
1,000
third
years
one up.
old.
Yeah, third one up.
amazing.
Wow.
But also very
So
slow
number-
growth
Yes.
as well.
four.
Right.
Which means that if they get damaged, it would take a long time for them to recover or grow back.
Hayden, is this sample 17? Roger. Roger, thank you.
I have 2.
There it is.
There we go.
Niskin fired.
Got it. That was- no, that was number 4.
Number 4.
Number 4.
Okay, what part of the plant do you want?
I didn't talk about plants. I would say the tip maybe, so somewhere around here.
Can you zoom in video?
Yep. Zooming in.
That thing is so convenient.
Makes things so much easier
To point.
to point out, yeah.
Uh-oh. I'm not close enough. I have to move the vehicle.
All right, going wide.
Where do you want to place that on Hercules?
I would say a forward bio box.
Would it have any issues with the long-stranded coral or the black coral that's...
I mean, we cannot mix them up. I think it should be okay. So that way we keep like some space in case we collect more rocks.
Okay, so you want those in the lambda box then?
In the same
Box
compartment.
with the other corals?
Yeah.
Okay, perfect.
I think that would be the last one we put in there.
Mm-hmm.
Cool view in the cinema cam.
Oh yeah. Oh,
Do you know if this is CITES or not?
Pan up. Pan up. Okay.
go ahead. Yeah, all good.
There we go. So you want it right around there?
Yeah, maybe a little bit like that over there. To get, if you can. Yeah.
Come on.
Nice.
Oh no.
Just I can't reach it. Do you want me to try to take another sample, or you want me to move forward and grab that?
Maybe try another sample.
Okay, pan up.
I don't want this is now rubbed on the. Let's try somewhere here, and otherwise if it doesn't work out, we can just leave it alone. There it is.
Nice, Danny.
Okay, bio box out.
Bio box coming out. Forward box coming out. Forward box is out. Tilting down.
Bring the camera in.
Okay, camera coming in.
I don't know how this is going to work, but...
It's actually a really big piece. Oh, and that bamboo coral is... Ish.
This is not working the way I wanted it to.
I think it's all right if we can get some of those pieces that are already in there.
Okay, close the box.
Closing box. Box is closed.
I don't know how well that's gonna work. That's gonna be in my camera the entire time.
It looks like it broke off, maybe.
I'm not gonna get my jaws that close to the camera.
Yeah.
Oh.
HD out.
HD out. It's extended fully.
Do you think this may have broken off when you close the box? Maybe if you reopen?
Yeah, we can reopen. Okay, I'm gonna bring the HD in. Yeah, I got it.
There were some little branches
Okay,
that
open
fell in.
the box.
Forward box opening.
Yeah.
Careful that bamboo coral.
All right.
Maybe
There we go. Good one.
Nice, Danny.
Yeah.
Okay, close the box.
closing.
Nice.
Box is closed.
All right.
Okay. We good here?
Yes.
Okay.
I'm gonna do a DVL reset.
Nope, I'm getting away from you.
Hmm?
Oh, I said I'm getting away from you.
Yeah. Small game. Still. And still come down for you.
Is that something in the sand?
It's one of these sea pens, similar to what we've seen earlier.
Very cool.
It's just so cool to see it all. Like it's like, you know, you see it on the screen, but then I can't imagine what it would be like actually down there. How huge this almost like drop off area would be, you know? Like I can't imagine what it would be like diving with one of those corals.
Yeah, it's always hard to visualize, especially in 2D when you're looking at it on the screen. Because we're looking at a 3D environment in 2 dimensions, so it's really difficult.
Exactly. Yeah.
To imagine.
I can't imagine. Like it has to be. Especially when you get into areas where it's just tons of them, you know?
Yeah, as somebody said earlier, the coral definitely does seem a bit sparse, you know, very spread out, but we do certainly have our dives where we see very, very high concentrations of coral.
Yeah, there's another Iridogorgia, as you can see right here.
Beautiful.
And then a bunch of other coral. Maybe we can look at this one here.
Yeah. Awesome.
I'm sorry, Fanny, I could not hear what you said.
Sorry. Can we zoom in on that little one here with the brittle star?
Yeah, no problem.
Thank you. We're going to leave the Iridogorgias alone for now. Yeah, they are really big, these colonies.
All right, so it is about time for the 4 to 8 crew to start wrapping up. We are going to switch out so that the 8 to 12 crew can take over. It's been really lovely hanging out with all of you for the past 4 hours. Thank you so much for joining in on the exploration, and we will see you again very soon.
Go ahead, video.
Hold on 1 second. Ready to zoom in.
All right, video zooming.
Zoom.
Sure.
Okay, pilot coming off comms.